are and what you've been working on.
My name's August, and I've been working on barcoding the DNA of fish that were collected at New York City farmers markets. Basically, we looked at one gene to see if they were all the same species, or how they related to the species they were sold as. We found that fish that looked similar to each other sometimes were sold under the wrong name, and it seemed to be accidental because there was no relation in between cheaper fish being sold as more expensive fish. In several cases, it was actually the opposite way. So it seems like people just can't tell these fish apart, and they're sold under the wrong name. We found one particular species of cod that was sold as 4 different fish. And since cod is considered a vulnerable species, their population has dropped by 80% in the last 20 years or so. This is important because it affects regulations. The EPA and other government agencies can't regulate how much of the fish is being caught if it's being caught and sold under different names. It also boosts the numbers for the other fish that it's being sold as to make it seem like there's more of these.
Can I ask you a question? What were some of the other names they were being sold under? Do you know the names of them?
Yeah. It was the species of cod was being sold under the names of scrod, hake, pollock.
Wow.
Those were the ones we found.
Those are very recognizable, so it's interesting that they were undetectable, at least visually, as being different.
Yeah.
Were you able to notice a difference as you continued to study, like just by visual?
We only worked with fillets, so
Okay.
every fish sample looks pretty much
Every fish looks
alike.
the same, and they all taste like fish, pretty much.
Yeah, so.
Good. All right. What else did you learn about this? What was the big takeaway that you found? Otherwise.
That was
the big one?
That was the big one.
I
We
think
also
that's
found
huge.
that many fish are labeled correctly.
Oh, really?
We found some fish that were what they sold as, even if a different sample showed that was really the species of cod.
Right.
And we found that somewhere along the line, one set of samples got contaminated with fish from Japan.
Oh, wow.
So,
That's interesting. Was there, in terms of the contamination, what form did it take?
all the samples came out as this Japanese anchovy, and we knew that all our samples came from fish that were more than a few inches long.
Right.
So we're not sure where it came from exactly.
Wow.
Came up with a few ideas,
That's interesting.
ranging from them being in with the fish when they were caught to the primers and PCR equipment we
Right.
used being selective for that genome,
Right.
or that gene, but I mean, we don't have the information to say for sure where it came from.
Wow, that's interesting. When you were selecting the different fish, which were because you said that most of them are labeled properly. I think you said more often than not they are. Was there a particular species of fish that you found that was labeled properly overall?
Yes. Skate is in every sample we had was a skate.
Every
species
time.
of skate.
Right.
And monkfish.
Monkfish.
Which we noticed that skate and monkfish are extremely recognizable.
They have very distinct features about them so that when you look at them you know this is a skate or this is a monkfish.
Right. You're not going to mistake skate or monkfish for a different type of fish.
Right, but the cod really is a chameleon, sort of.
Yeah, it seems to look
Like a lot
of other fish.
So we can call it the chameleon cod from now on. Anytime you walk through Chinatown and you're like, that could be a codfish, no matter what it's called, right?
Yeah.
Great. All right. Was there any other information that you wanted to share with us about your findings?
Well, I I guess I'll explain just after you extract the DNA from the samples, you put in a primer with the DNA that's being extracted, and that isolates the gene that you want to look at. So for each gene, there's a different primer that's used. Otherwise, you'd multiply the whole strand and get 2,000 or 10,000 base pairs, which is too much to look at. That's it.
That's it? All right, let's give him a round of applause. So for you, the next time you're in Chinatown and you're walking through the fish area and you're like, I want to have some skate, you're going to get skate. But if you choose something else, it could be cod. That's pretty much what I figured out from what you're saying. Good, really good work, really interesting.